All right. So yes, I was assigned to be a moderator as any early stage researcher of ALLODD. We are given this opportunity to present the speakers of ALLODD webinars. And so right now it's my turn, and I'm lucky because this is Jean-Pierre Changeux with us today. So let me give you a brief introduction of Jean-Pierre Changeux' career. So he is an emeritus professor at the Collège de France and Pasteur Institute Paris, and also international faculty at Kavli Institute for Brain and Mind at the University of California, San Diego. And his main contributions and discoveries in the course of the past 50 years are centered on the general theme of receptors and their allosteric transitions, primarily in the nervous system. So these contributions were initiated by the first identification of a neurotransmitter receptor, which was the nicotinic acetylcholine receptor. And then Jean-Pierre combined approaches from pharmacology, molecular biology, developmental biology, as well as behavioral and pathology approaches to demonstrate that the nicotinic receptor is a bona fide allosteric protein. Then he further elucidated the molecular mechanism through which drugs modulate receptor efficacy when binding to distinct allosteric sites, thereby opening a new avenue in the field of drug discovery. From my side, I would add that Jean-Pierre Changeux is a legend in computational drug design for synaptic receptors subfield, because we do use mono-Wyman Changeux model to describe thermodynamic equilibria between different conformational states of allosteric receptors, and that's a formula with his name. And so just to give some details on another contributions and on the career. So in terms of the timelines, Professor Changeux was trained at École Normale Supérieure and the Institute Pasteur, and then received the PhD at Paris University in 1964. And then he was a postdoctoral fellow at the University of California, Berkeley, from 1965 to 1966. Then visiting assistant professor at Columbia University at College of Physicians and Surgeons in New York from 1966 to 1967. Then sous-directeur Collège de France in Paris, also director of the unit of molecular neurobiology in Institute Pasteur from 1972 to 2006. At the same time, professor of Collège de France from 1975 to 2006, professor of Institute Pasteur from 1975 to 2006, and emeritus since 2007. Then last two things is that he was also a SCAGS distinguished visiting professor in pharmacology at the University of California, San Diego from 2008 to 2012. And finally, international faculty at Kavli Institute for Brain and Mind, University of California from 2012 to 2022. But what is interesting is also that Professor Changeux has authored several books for a non-specialist audience. So notably, Neuronal Man, The Biology of the Mind in 1983 to 1985. Then Conversations on Mind, Matter, and Mathematics with the mathematician Alain Connes in 1998. Then What Makes Us Think with the moral philosopher Paul Ricoeur in 2002. Also Physiology of Truth in 2005. And the Enchanted Neurons with the musical composers Boulez and Manori. And also there is a recent interview, well, in French it's called Le Beau et la Splendeur du Vrai, which was made by François Yvonnet in 2023. And of course, Jean-Pierre Changeux has significant amount of academic accolades, which include the Gardner Foundation Award from 1978, the Wolf Prize from 1983, the Louis Janet Prize for Medicine in 1993, the Baltzan Prize in 2001, then the National Academy of Sciences Award in Neurosciences in USA from 2007, the Olaf Thon International Research Award in Biomedicine obtained in Oslo, Norway in 2016. Then Albert Einstein World Award of Science in Hong Kong in 2018, and Clarivate Citation Laureate in Physiology and Medicine for 2021. I was also a witness of a special emeritus professor status at Urbino University, which Jean-Pierre was obtaining just recently, 1 year ago during the ALLODD meeting in Urbino. So anyways, thank you very much, Jean-Pierre. The floor is yours. I am very interested in your talk personally, and please, we are really interested to hear what you are to say here today. Thank you very much.
Okay, thank you very much. Thank you, Gregory, for these very just words. I hope I am not going to disappoint you after all of these decorations, if I may say. So I am going to, in fact, present to you a general view about the contribution of Allostery in drug discovery. Of course, I am not going to reinvent the history of Allostery, but nevertheless, I have to mention it if I want to try to address some critical question of drug discovery, which I will do at the end of my presentation. So for those who know part of my presentation, please be patient until the end, where perhaps there will be new things for you. So the presentation will include some fundamentals about allostery, as I just mentioned, the concept of allosteric modulation, then the issue of conformational transition of allosteric protein and conformation-specific drug design, then point to allosteric disease, which I did not do it so frequently. And last but not least, I will try to approach what I call precision pharmacology, which is, of course, a complement of precision medicine, but from the allosteric and pharmacological point of view. So the issue which I am going to discuss comes from Pasteur. As you heard, I have been working at the Pasteur Institute for many years, and the concept of chiralité, which was introduced by Pasteur, and related the stereo-specific interaction. Now, as you may know, when he was a student at the École Normale Supérieure, Pasteur discovered chiralité and separated with his hands, right from left-handed, crystal of tartaric acid in a mixture which was inactive from a rotational point of view, and he found that the right-handed dextro rotatory and the left-handed levorotatory modified polarized light according to this. So this created an issue that there would be some kind of stereo-specific organization of these organic molecule, which would have some selective action on living organisms. So he applied the concept to fermentation, and by chance one day realized that in a racemic mixture of tartaric acid, after the growth of some bugs, then there was only one of the two isomers which was used by the organism. So it means that yeast, which were of course concerned as well, degrade right but not left tartaric acid, and this was also applied to taste different on taste receptor, and therefore he made a very strong statement, which is still very true for us, that the active dissymmetric body which intervenes in the nervous impression is the nervous matter itself. A dissymmetric matter, as all primordial substances of life, And this has been, of course, this was confirmed with the structure of proteins that were, as you know, on a monomer of protein, then there is an asymmetric binding site which possess a stereospecific relationship with the ligand. So this was really a fundamental issue, which was exploited on the drug design side by Daniel Beauvais, who was working at the Institute Pasteur in the 20s and 40s, and with his team discovered many compounds which are active on either bacteria or the nervous system. The thiaminophenylsulfamide as the first bacterial antibiotic, first sympatholytic versus sympathomimetic drugs, which are structural analog of adrenaline, first antagonist of histamine, which is also an analog of histamine, and in 1946 he designed flaxedil, the first synthetic Curie, and succinylcholine as well, which both act at the receptor at the of acetylcholine at the neuromuscular junction. Very interestingly, all these drugs are structural analogs of neurotransmitters. So he developed some kind of a rule about how to get drugs for therapy, and he proposed that the origin of many drugs must be looked for in substances of biological nature, and that in each case there is a physiological property of the compound seem to be similar to the compound synthesized itself as a drug. So the conclusion is that we should, I'm sorry, there is something which happened. I don't have my screen anymore. Okay, do you hear me?
Yes, yes.
Okay, so he concluded in his Nobel lecture to draw your attention to the importance which the study of competitive phenomena assumes in pharmacodynamics, and in particular in the pharmacology of the autonomic nervous system. So he developed what may be called what's maybe, sorry, there is something which is not, I'm sorry. Okay, so he developed the notion that competitive pharmacology is a main paradigm in drug design, and I may say since then this is still a fundamental notion, in particular in the industry, where this analogy or homology between the drug and the natural compound is still present in the mind of almost everybody. Now, this is something which are going to nuance by the idea that one might design drugs which are not competitive ligand for some biologically active sites, but that some additional kind of ligands may play a fundamental role. So this is why I introduce this presentation by the fundamentals of allostery. And you have to realize that we are here with bacteria in a situation which is very fundamental for the understanding of life itself. There is in a living organism biological regulation which is taking place, and in particular feedback regulation, which were already mentioned by Norbert Wiener long time ago. And so to understand life, you have to realize that biochemical reactions are not independent one from the other, but that they are cross-regulation between different biochemical pathways. And this was actually discovered by Humberger in 1956, that in the biosynthetic pathway of isoleucine, the end product of the pathway feedback inhibits the first enzyme, which is threonine deaminase. So we are here with a typical feedback mechanism at the molecular level taking place on a bacterial enzyme. And in addition, very interestingly, this feedback is in fact sensitive to a threshold concentration of the feedback inhibitor. And therefore, they are, in addition to effect between different molecules, between isoleucine and threonine, they are cooperative effects which are taking place. We shall come back to that later on. So when I started my thesis work at the Pasteur Institute, I was very interested by this notion that there is a specialized mechanism involved in feedback inhibition. And immediately I saw that there was some kind of structural feature in the molecule of the threonine deaminase, which was responsible, engaged into this feedback mechanism. So if there was such a mechanism, one should be able to uncouple it. And indeed, I did that with threonine deaminase through short heating times. And you can see that the control where there is a feedback by isoleucine is leading to an enzyme which is still active, but not feedback inhibited by isoleucine anymore. So there was an uncoupling of the regulatory mechanism involved through the feedback inhibition by isoleucine. So the same finding was also found in parallel by Gerhardt and Pardy with aspartate transcarbamylase. And I was at the time a graduate student of Jacques Monod, and he asked me to make a presentation at the Cold Spring Harbor Symposium on Quantitative Biology in 1961, of which he was the chairman there. And then for the first time, I presented in public this data and proposed, this is from the original paper, that there might be possibility, first of all, the classical model of competitive interaction I just introduced already in my first slides, and an alternative one where, in fact, the site for the substrate and for the competitive, for the feedback inhibitor, substrate feedback inhibitor, were topographically distinct. So this was an interesting concept, and at that time I used the word no overlapping site. And in the conclusion of the meeting, Jacques Monod came back to my findings and model and proposed with François Jacob the word allosteric. And I may say that the concept have been vehiculated by the word allosteric in much more efficient way than no overlapping, which was a little bit too common. So this is the birth date of the word allosteric with two stereo-specific sites. And of course, the interaction has to be mediated by a change of the protein, which carries the two sites, and the conformation change. Throughout the work I did in my PhD thesis, I was able to analyze the relationship between, first of all, the heterotropic effect of some of the allosteric ligands. I discovered some allosteric activator of the threonine deaminase, and to show, and others groups as well, that there was some relationship between the cooperativity of the response curve of the normal enzyme, and after desensitization, that there was also an uncoupling of the cooperative interaction. So this aspect of the cooperative interaction was, of course, in relation. I actually had the opportunity to meet Max Perut in the 60s, soon after I made this first paper, and he was, of course, extremely interested. And his opinion was that to account for the cooperative effect, then one has to, of course, relate the cooperativity with the organization of the protein molecule. And in the case of hemoglobin, there are, as you know, 4 subunits which interact together and give rise to this cooperative binding curve. So he had already, it was on the way to publish the structure of hemoglobin, which is indeed a symmetrical molecule, thanks to the fact that the protomers or the monomers are regularly organized in symmetrical structure. So this led us to propose with Jacques Monod and Jeffrey Weimann a model which was proposed at that time simply to account for data I just illustrated before, which I obtained in my thesis work on the relationship between cooperativity and the effect of regulatory ligand. And I had made some observation during my thesis I cannot discuss here, which led us to propose that, in fact, the simplest way to account for this very complex effect was to propose that there are 2 states of the protein, and that this protein is an oligomer where the relationship between subunits are modified, and where the affinity for the ligand, for the activators and for the inhibitors, differed in one conformation and in the other conformation. So this is the idea of a 2-state model, which postulate that the 2 conformations are in equilibrium, and that signal transduction or feedback inhibition would, for instance, start from a situation where this active state is present in large amount, is dominant in this equilibrium with an equilibrium constant of L, and then that the inhibitor shifts the equilibrium in the other direction. So this is one of the main issues of the model, that one postulates for, in fact, a single effect, that of feedback inhibitor and the cooperativity. In fact, a transition between 2 oligomeric states, which is in fact mediate the signal transduction mechanism. So this was formalized, as you see here. And what was very interesting in this scheme was that one could distinguish a state function in the sense that we can, for instance, monitor the active state compared to the 2 states, and a binding function, which is the actual binding of the ligand. So you can already see that from this picture that the binding function is different from the state function. And this is, of course, an interesting proposal that, in fact, if we have a built-in molecular switch which account for the feedback inhibition, then one should be able to test for this hypothesis experimentally. And I may say that this kind of scheme with pre-existing variation of the conformation, the variability of the conformation, and the selection by the ligand may be related to some kind of Darwinian mechanism. But the interesting thing I may insist in this presentation is that, of course, there is a difference between the most evident, simple-minded kind of concept, which is largely used, unfortunately, in drug design, still in big pharma, that in fact the ligand, when it binds to the site, induce the change of conformation. So instead of selecting one of the two states, the ligand, in fact, just go onto one state conformation and then modify the conformation of the protein when it binds. So there is some kind of steric fit, and the conformation change occur only after substrate binding. And this is, of course, a very important issue. And same thing for the cooperative effect that you see on the right. And the process, I may qualify something like a Lamarckian process, which is the induction of the shape by the actual action, and from the selective Darwinian mechanism I illustrated before. So this important point is still debated, of course. There was a lot at that time, and it actually lead to a direct distinction between the two states by measuring separately conformation and binding, which I did in the laboratory of John Gerhardt and Howard Shachtman, and where the conformation can be followed by sedimentation or by fixation of diol group reagents, whatever. And when you compare the state function from the binding function, they are clearly different. So this, of course, could be fitted by the model. And there are, of course, other experiments, and I will try to illustrate them now in my presentation, which are in agreement and consistent with this two-state model. And of course, how to test the model. And in my thesis work, I wanted to get out of the bacteria and go to the brain. And I proposed the extension of the concept of allostery to brain communication. And you all know that there are neurotransmitters involved in chemical transmission at the synapse in the brain, in the nervous system in general, and that this neurotransmitter is acting on a receptor. And there are now more than thousands of receptors. And for a number of neurotransmitters, which is still rather limited, as you see 12 small molecules that all of you know, from acetylcholine to GABA and glycine, glutamate, and about 100 neuropeptides, as was recently, in the past decades, developed by Thomas Krell. So we may view the brain indeed as a highly diversified chemical machine. And if we want to test allostery, of course, we have to go to a well-identified single molecule of neurotransmitter receptor. And at that time, they did not exist. So this led us to identify the receptor for acetylcholine through electric organ of fish and zinc venom toxin, and have the purification of the receptor done with the electric fish electrophorus. And you can see here in the postsynaptic membrane some layer of receptor just below the nerve ending. And you can already see that these are pentamers, and this was largely developed by Carlin, Raftery, and together with Hryhory at the Pasteur. Then we identified the first amino acid sequence, and several of them were done later on by Heinemann, by Numa, who developed extensively systematic analysis of several subunits and several receptors and the full amino acid sequence. And the model that all of us propose in 2 dimensions was there was an extracellular domain which carries the acidic binding site and a transmembrane domain involved in the ion channel. And that was some kind of very simple-minded model. And the question came, of course, immediately, is the receptor an allosteric protein? You may already see that on this scheme, there is a first indication, since, according to the scheme hypothetical at the time, the acetylcholine site would be located at a different domain of the receptor. So that was the beginning, and this is the end, if I may say, because there is a paper by Marco Cecchini and Pierre-Jean Coronet and myself in Annual Review that we published this year. And you have here the high resolution by X-ray crystallography and electron microscopy of the nicotinic acetylcholine receptor. You recognize the synaptic domain, the transmembrane domain, and the cytoplasmic domain, and also the site for acetylcholine and for nicotine in green here, and through the transmembrane domain that these alpha helices, which 4 types, and the M2 type lines the wall of the ion channel. So clearly, there is a topographical distinction between the acetylcholine nicotinic binding site and the ion channel. And the distance was actually measured by carefully by fluorescence spectroscopy by Palmer, Taylor, and others. And clearly, it is there is a far distance between the 2 categories of sites, and therefore we are dealing with a bona fide allosteric protein. So this is important for us for drug design. I may insist on that, we may have sites, not only at the level of standard biological sites, which would be nicotine, but also channel, and as you shall see later, other places throughout the nicotinic receptor protein. So the question, of course, was if this is true for the nicotinic receptor, is it true also for other receptors? And fortunately, we were able, throughout our studies, I'm coming back earlier, to a stage where we were unable to get the structure from the nicotinic receptor from electric organ. And there was a publication by Tasnim who found some sequences in the genome of some bacteria which resemble those of the nicotinic receptor. And we and Itman in parallel looked for some receptors, homologues of the nicotinic receptor, in the bacteria Glic and in the bacteria Elic. And the very interesting thing is that indeed we were able, Pierre-Jean and collaborators, to purify, measure the X-ray structure of these receptors from bacteria. And following this work, Guo, with the glucel from the worm, and then Miller and others from GABA receptor, glycine receptor, the serotonin receptor, and most of all by Noviello and collaborator for Ryan Hips, from the University of Cala San Diego for the nicotinic receptor in these different organizations. So this is, I think, a very interesting situation, because we have here, in fact, in front of us, some critical receptors from the brain and for several neurotransmitters which are different from each other. And in fact, the receptors are highly homologous. And in addition, they are also homologous from receptors from which they originate in bacteria. And this, of course, is a very interesting situation for us, where evolution have in fact preserved the pentameric structure of these very original receptors from bacteria to the human brain. And of course, we have to realize that our brain works with receptors which are operating like or similarly to those of bacteria, therefore, not at the speed of light, like the computer, but at the speed of sound. So that means that this is the reason why, if I may say, our brain receptor and our brain is so slow. So this conclusion is of interest if you want, in fact, to study the pharmacological and physiological properties of these receptors. And indeed, as I already alluded very quickly, there are, in addition to the neurotransmitter receptor binding site and to the ion channel site with competitive blockers, which are present for each of these sites, other category of sites which are present in the synaptic domain, in the transmembrane domain, and in the cytoplasmic domain. So we have binding sites, which are illustrated here, which for these different domains, which in fact modulate the activity of the receptor and modulate the signal transmission mechanism, here the opening of the ion channel. So we have here, from a principle, interesting situation where these allosteric modulatory sites are topographically distinct from the orthosteric site and can bind ligand highly specifically, as we shall see in a minute. And this is, of course, extremely important because, according to Daniel Beauvais, competitive pharmacology, here we have an analogy between the drug and the neurotransmitter, and there might be some interference with brain synaptic. Here we don't. These side bind compounds, which are completely different from the neurotransmitter binding site, and there is a new pharmacology, therefore, which is opened by the study of these allosteric modulatory sites. Just an example, ivermectin, which was discovered by Omura and Campbell to be a very efficient anti-helminthic, and here we discovered with Daniel Bertrand that if we add ivermectin to the alpha-7 nicotine receptor, we found a dramatic activation of the receptor. You see the dose response curve here in the control and in the present time. And we also showed that if we mutate the transmembrane domain, then the response to ivermectin is no longer present. So we have here a very interesting site, which is located in the transmembrane domain. And here I showed you the data from Hibbs and Guo, and you see here in yellow ivermectin. Here you have the synaptic domain, here the transmembrane domain, and here you can see at the interface between M2 and M1, the molecule of ivermectin. Here an overview of the binding site and of the molecule within these alpha helices, which compose the transmembrane domain. So we have an allosteric binding site, which is not a pocket, if I may say, but an in-between transmembrane alpha helical site, asymmetrical, like Pasteur would say, and therefore, from this site, we can control the activity of the receptor, and this was shown further by Hibbs and Guo, that in fact this effect of ivermectin on the glucial receptor from the elegans is responsible for its anti-helminthic action. So this is not a simple case, and since this early work, many allosteric sites have been discovered with the nicotinic receptor family, here with the glycine receptor, and you can see that there are six categories of allosteric sites, and you recognize among them pregnenolone, which is an hormone, picrotoxin, and tetrahydrocannabinol. So you have an ensemble of allosteric binding site, an allosterome, if I may say, of the receptor protein, and you have the same thing, and this can be, of course, generalized, as I shall say in a few moments, with the nicotinic receptor and some binding sites in addition to the ivermectin binding site. For those interested, this database has been published by Cerdan et al. And also in this Unistrasse. So to really understand signal transduction and the effect of allosteric modulators, which make the whole very complicated, in fact, one simple way to interpret the effect is to come back to the two-state model. So we did that with Thierry Edman a long time ago, and by using fluorescence spectroscopy and stop flow, we identified several relaxation times. And I'm not going into detail, but from these studies, we proposed a scheme which is, in fact, at this stage, quite significantly more complex than the two-state model. We have indeed the two-state model, which is satisfied, and which is also strongly supported by the fact that Jackson discovered spontaneous openings in the absence of acetylcholine. So if you have an opening of the channel in the absence of the neurotransmitter, it means that the states are pre-existing. So this is consistent with the pre-existing story. But the interesting thing is that we were able to show that there are other relaxation times to conformation transition, which are consistent also with some physiological studies which were made long time ago by Katz and Teslev, that in addition to the resting active states, we may have a desensitized state where the transition is slow, and an intermediate state. I will demonstrate that later on in my presentation. So, okay for the two-state model, but there are more than two conformations. And again, this is even a further demonstration of the selective model, since we can here have ligand which selectively bind to these different conformations, as I shall show in a minute. Briefly, the conformational change involved in the allosteric transition between, let's say, the resting and active state. We are considering only this two-state transition at this stage. You can see that there are changes which are taking place in the conformation between Glick, which is the open and closed conformation. And this was done by Marc Delarue, Pierre-Jean Coringet, and their team. And we were able to show that there is some kind of twist which was taking place, which link the synaptic domain with the transmembrane domain. And there is also some kind of opening, we call it a bloom. And this was further clearly demonstrated by molecular dynamics by Marco Cecchini, and who was a collaborator of Martin Karplus and became independent at that time in Strasbourg. And you can see that one could, through molecular dynamic simulation of about 200,000 atoms, which is an achievement at that time, the molecular dynamic simulation could be, to a large extent, obtained for this switching transition between the racing and atomic and open state at the atomic resolution. So this again impose dynamic constraint on the brain receptor, well, I already mentioned, because this was done with the bacterial receptor, and these studies with the eukaryotic receptor, BCL, and same thing with the brain receptors in humans. So this molecular dynamic here control, in fact, the mnemonic of the physiology of our brain receptors, and has a critical role, of course, on cognitive functions. So this allosteric transition of activation has been also studied with by other groups like Hryhory, and according to our recent views, this was done by structure and also by molecular dynamics. Here you have the mechanism through which the link between the orthosteric side and the ion channel is being established through some kind of twist and tilt, which you can realize here. So there is a twist and tilt which take place during the signal transmission between the synaptic domain and the transmembrane domain. So this was further analyzed by Pierre-Jean Coringet and shown to take place progressively through the molecule of the receptor from the synaptic to the transmembrane domain, and there is a progressive change, which is like a conformation of the wave, which is taking place between the synaptic domain and the transmembrane domain. Now, it's time to examine the case of desensitization. If you remember well the model with Thierry Edman that we proposed in 1981, there was the proposal that in addition to the resting and active state, there are states which are desensitized, which were referred here on RND. These states were actually observed by high resolution cryo-electron microscopy with the alpha-7 nicotinic receptor. You see the 3 states here. And Marco Cecchini and Maria Astrikova were able to make molecular dynamic simulation together with Markov state stimulation. And here to illustrate the pore radius of of the active state, of the resting, and of the desensitized. And you can see, of course, that here the size of the pore, 2 angstrom, is very small, so the state is inhibited. There is no transport taking place. There is transport actually taking place in the channel, and now in the resting there is no transport. I'm sorry, just to be clear. Here is the active open, and you can see here the pore between 4 and 6. Here resting closed, the pore size 2 angstrom. And the interesting situation they discovered is that when we may have in the desensitized state not only closed, as expected from the early model of Katz and Teslev, but also opens. So that was, in fact, a discovery that there are desensitized open states which can be represented very simply here of the scheme, that you have the resting active and the desensitized and something else where the channel is open while here it is closed. So this is a 4-state model consistent with the one I showed here, but much more precise and specific, of course, than in these early studies. Now, interesting, this kind of situation take really an importance for drug design, since going back to ivermectin, you remember I mentioned ivermectin as a positive allosteric modulator. So it's causing an increase of the response. And there are other types of PAM, which we call type 2 compared to ivermectin type 1, where there is also an effect, very strong effect, shoulder on desensitization. Here, desensitization is very fast. It's a very transient response, which is amplified by the type 1 positive allosteric modulator. And the cryo-EM structure reveal that, in fact, there is indeed a very interesting situation where the type 1, the ion channel closed normally, while in the type 2, there is some kind of opening which persists here. There is some state that we may refer to as desensitized, where the channel is open. So, in fact, we have 2 types of allosteric modulator on the same receptor, and some give pure potentiation of the response, like the type 1 ivermectin, and other stabilize open state with a characteristic come from a pharmacological point of view, different from those of type 1, and of course the resting and active conformation. So we are now in a situation where we have, in fact, not only the agonists and antagonists in the 2-state model, but in this 4-state model, now we have positive allosteric modulators which are active either on the active or on the open desensitized state, and at equilibrium on that, and then others acting on the desensitized conformation. So we have here with PAM1 and PAM2 some conformation-specific allosteric modulators, and this is of course known from the resting and active, where you have indeed a conformation-specific for the active or for the resting, but in the modulation, this is something really very new and quite interesting for pharmacology and drug design in general. Now I am going to shift the next few slides, briefly, before showing some concluding remarks, about a mutation of the nicotinic receptor. And of course, this is of clear importance if you want to design drugs against a disease. And many diseases are due to mutation of receptor. Not all of them, but some of them. And we curiously make a discovery with Daniel Bertrand, very unexpected, at the time where this is, you can see in the 90s, when we were studying the amino acids lining the ion channel in M2. So we mutated some highly hydrophobic amino acid like leucine here, the leucine 270. And we were thinking that if we were going to mutate these molecules, we would block the ion channel or have no block the response and so on. And I remember Daniel Bertrand called me, oh, I have a very, very unexpected result. I have more activity than in the controlled wild type. How do you explain that? And this is indeed a very interesting situation where these mutations here, indeed give a receptor which is much more active because you see the shift of the dose response curve and have also a lot of pleiotropic effects, loss of desensitization, a new pharmacology, and so on and so forth. So that was really a paradox for us to realize that the receptor should be able to undergo such kind of conformation transition, giving a hyperactive receptor. And as you know, it's sometimes better to find a drug which is activating the receptor when it is missing. But here we have to find a drug which would block the receptor to be efficient. So this was, in fact, an interesting discovery, which was, of course, discussed by and documented further by other groups, in particular the group of Berkowitz in Australia. And he was studying autosomal dominant nocturnal frontal lobe epilepsy. And you can see here the poor child having this convulsion in the bed, waking up during sleep. And very interestingly, Berkowitz discovered that the mutation which give the epilepsy are exactly at the same place in the transmembrane domain as those we showed initially with the so that was a real confirmation, a very strong confirmation of the finding that some mutation can give a hyperfunctional receptor and here cause a disease. So this, of course, needs a particular pharmacology since we have to now try to block the open channel conformation. And since the mutation we are having here is in fact stabilizing the active or an open desensitized conformation, as you shall see later. So this is, of course, an importance for the pharmacology of not only epilepsy, but disorder of conscious processing in general. But meanwhile, the group of Sine and Auerbach and others have found mutation, or discovered the mutation in muscle nicotinic receptor, which can cause loss of function, which is more attended, expected, but also gain of function. And this gain of function also lead to a blocking of the synapse because it's inactivate the receptor through calcium entry into the postsynaptic domain. So these mutation gives weakness to the eyelids, and very interestingly, they are located not only in the transmembrane domain. You recognize here in green the gain of function I just mentioned before with the epilepsy receptor. And the interesting question we raise, since these mutations are also present in the synaptic domain, and the two kinds, also in red here and there, you can see. So what is the mechanism of this mutation? And in a work done by Antoine Tally with Martin Karpus in Strasbourg, it was discovered that in fact these mutations are located at the boundary between subunits. So that's also something of interest, because it means that the twist and bloom that I mentioned before indeed affect the relative position of the subunit into the oligomeric structure. And here that we have a quaternary change, a quaternary twist in the molecule, which is responsible for the change of property of the receptor, not only of the protein, but of the sites which are carried by the receptor itself. So this is, of course, of importance, and it even became more interesting. This is a very recent work of Hryhory and Marco Cecchini, that they were able to examine with the alpha-7 nicotinic receptor the mutation that, as I illustrated before, give this hyperactive receptor L247T, if you remember. And here is a profile of the ion channel, and here again the white tap close to angstrom, here 4 open. So we have, in fact, a desensitized open state. You heard about it. I mentioned it. This 4 state, resting, active, desensitized, intermediate. So the very interesting thing is that, in fact, one can find that the mutation, in fact, which creates the disease, stabilize the open desensitized state, where, of course, the channel open, and which gives the pathological phenotype. So the pharmacology then has to be adapted to the type of conformation which is altered by, stabilized by the mutation. And therefore, if you want to make a precision medicine, you have to relate the genome with the disease. Here, I may introduce the word of precision pharmacology, which makes development of pharmacology, which is adapted to the receptor phenotype associated with the mutation, and more specifically, the receptor conformation, which is associated with the mutation. So this is a really very early stage, and it has, of course, to be further developed in the future, but it's where we are today. Now, I will close with the future of pharmacology. First, I talked about organic compounds, as Daniel Beauvais showed at the very beginning, but there are also other kinds of compounds, and in particular antibodies, which have been very popular those days. And here, the work of Pierre-Jean Changeux, who developed antibodies against, again, alpha-7 nicotinic receptor, extracellular domain. And here is the location. Here is the extracellular domain, is the location of the antibody, as shown by high-resolution electron microscopy. And he was able even to show, very interestingly, that the nicotine is still binding there. So the effect of the antibody is an allosteric effect. It's not blocking the active site itself, but changing the conformation. And you can see here that one of them, the E3 PAM, is the PAM, which is a positive allosteric modulator. The other one here, the C4, is a NAM, which is in fact blocking the effect of the E3 PAM. So we have now, with allosteric nanobodies, a new class of nicotinic drugs, which has to be further investigated in the future. Maybe we have a new pharmacology, which is not an organic pharmacology, but nanobody pharmacology for the future. Just to illustrate also that lipids that we showed very early on to torpedo receptor, that was done with Vandin in 78. And here the recent work of Bazak and collaborator, which shows that membrane inhibitors can play the role of allosteric modulator in channel gating at late stage of the conformation transition. So this is again new pharmacology of endogenous ligands, and as I mentioned, tetrahydrocannabinol anandamide. Anandamide is the physiological neurotransmitter, are acting as an allosteric modulator of the glycine receptor. So this is just to illustrate the immense diversity of the allosteric modulatory site on different kind of receptor, and from ligand-gated ion channels, GPCR, tyrosine kinase, and even transcription factors. And just I will close on 2-3 slides to illustrate the case of muscarinic receptor. As some of you know, there are two main categories of receptor for acetylcholine: the ligand-gated ion channel and the GPCR. The muscarinic, GPCR, versus nicotinic receptor, I just discussed throughout my presentation. Now, thanks to the work of several groups, including Kubica, when is able to show that there are indeed allosteric sites on the muscarinic receptor. Here, you can see the in yellow the orthosteric site, and in violet the PAM. And very interestingly, there is work going on now on orthosteric ligand of the M4 muscarinic receptor, and Novo Nordisk have discovered a very potent neuroleptic, which is a cholinergic neuroleptic acting on the muscarinic receptor. This is a revolution because in the past, most of them were again dopamine, and that's a new kind of neuroleptic. And you can also see that recently there is a positive allosteric modulator of the MM4 receptor, which has been identified and which is under clinical assay. So we are here with a new category of antipsychotic action, which is, of course, very important for the pharmacology of the brain. Now, there are also sites, allosteric sites for GPCR, which are of importance for morphine anti-situation and withdrawal sign, which have been discovered by Kubica and others. And very interestingly, the development of tree therapy, which is very efficient for cystic fibrosis, which goes CAF-trio. And in fact, there are 3 compounds, which are, 3 of them are allosteric modulators. The first 2 acting on the folding of the protein, and the other one as a typical allosteric modulator, which is binding to the transmembrane domain of the CFTR channel. and therefore the actual tree therapy for cystic fibrosis is allosteric therapy. So this is really to show the importance of allostery in drug design. Now I will close with one other kind of receptor, which is the transcription factor. One can actually demonstrate that they form heterodimeric molecule, and that there is a ligand binding site, a DNA binding site, which are different, of course, and that they are developing compounds which block the heterodimerization. And this was done by Igor Sigelny, essentially, and also in collaboration with the late Rudolf Rigler. One can show that some of these compounds do separate the two monomer of the heterodimer, and these compounds indeed have a very strong pharmacological action since they block the glioma cell growth. So there is a new opening here in the pharmacology of gene transcription and allosteric pharmacology of gene transcription. This is also applied to toll-like receptor. And just to close with saying that there are about 91 allosteric modulators available and used as medicine, plus the conformation-specific monoclonal antibody, which are used with cancer. And my take-home message is that we started from the concept of allosteric interaction between topographically binding sites. We propose a two-state model, which happen to be okay, but not sufficient, because there is allosteric modulation and the development of demonstration that in a receptor there are more than two states, and that one can have at the atomic resolution a conformation-specific drug design. And last but not least, that for allosteric disease, one can develop now what I call precision pharmacology. So there is a future for allosteric pharmacology. And I may mention the recent Pasteur Institute and University. It's interesting that here Pasteur is the link between the collaboration of the laboratory of Marco Secchini and Pierre-Jean Changeux, and emphasize the fact that this work, the late work, since I am now with a certain age, was nevertheless supported by HBP, which gave me the possibility to have student and postdoctoral fellows. Thank you very much. Thank you, Zoe, for this invitation to talk about allosteric proteins.
Thank you so much. Thank you so much, Professor. I think audience may have questions. So if anyone has any question, please raise your hand, or because I have also something to ask. But would anyone else want to start? So, I mean, if not, maybe Julia. All right. He just turned on your camera. So, all right. So, I wanted to ask you first of all about one recent issue which we have been discussing here at J&J. So, I'm doing a second here right now. And you see, we have looked into the orthosteric binding site of glycine receptor when it is in closed and in open state, especially in cryo-EM structures. So let's say with taurine or with GABA, where they claim that they have captured open and closed state. And you see, the issue is that on the level of the orthosteric binding site so on the level of the first shell of the ligand and its surrounding residues, we don't see too much difference between these two states. So the only thing which is really depicted in deposited cryo-EM structures is, first of all, that depending on closed or open state, the size of this orthosteric binding site is somewhat different. So it may be a little bit shrinked, it may be a little bit enlarged, but there are no, you see, radical conformational changes seen in the residues of the first shell and something of that way. And so we are just asking ourselves, and also people from J&J were asking me, how come that we don't see such a big changes in the orthosteric binding site, while on the conformational level of the protein, we see a big change. So the pore, it can be closed, it can be open. We do see these twisting and blooming aspects which you were mentioning in your presentation. And the question is, so can it be that on the orthosteric level side, binding site level, can it be that when it comes to closed or open state, the difference is maybe dynamical? Maybe if we sample closed state and open state, maybe we will see, let's say, more often that the C loop is opening maybe in the closed state rather than in open state. So we are really not sure why there is no so much difference between them on the binding site level. So my question, like, let's zoom in to the binding site.
I understand. Very well. My first response is that the techniques are not good enough to show the difference. And the main problem with the active conformation is that it is a transient conformation. You have to realize that. It's easier to have a resting or desensitized conformation on a plate to measure the to follow the structure. And my view is that it is a dynamic structure, and that you have to capture it by dynamic techniques. So I would suggest, if you want to see it, to develop something which lets you see it. Now, of course, the molecular dynamics may help you, and I'm pretty sure that the molecular dynamics show a difference between the resting and active conformation. So the fact that you don't see it does not prove that it does not exist.
Right, all right.
Sorry to say that, but it has always been for us, even from a biochemical point of view, an issue to isolate and identify the active conformation compared to the resting and desensitized. So as again, it's a very transient kind of conformation. Mm-hmm. It's a very fast one, and it has been selected for that, I may say. Because if the affinity and the stability of the active state was too high, then the synapse would not work in a repetitive manner.
Mm-hmm. Yep. And a second question I had was in relation to, because you were talking about allosterone. So you were showing these pictures with many possible allosteric binding sites, both in glycine receptor and in nicotinic acetylcholine receptor. So my question is here very general. If you go back to this picture, can you sort of tell whether non-transmembrane domain binding sites, which ones of them are the most interesting in your view for drug design? And why I'm asking this question is because, well, it's for my personal interest, because we do, with Marco Cecchini, we do computational simulations, computational drug design ideally. And we do understand that to try to calculate binding affinity in transmembrane domain, especially since, as you mentioned, there can be competition between the lipids themselves, it may be too ambitious right now. However, any part of the protein which is outside of transmembrane domain, be that intracellular or extracellular, it is totally possible. So we do know very well about orthosteric binding site, but which ones from your sort of experience, from your vision, are the most interesting to target, both in glycine and in acetylcholine or nicotinic acetylcholine receptor?
Initially on the structure of allosteric site in the transmembrane domain, we published a paper on general anesthetics with Pierre-Jean Changeux. And this is, of course, an interesting situation because, as you know, you block the activation without acting at the level of the orthosteric site. So we showed that it was at the level of the transmembrane domain, that the site was, through mutagenesis and other things, and we proposed exactly what you said, that in fact the site of action was the lipid binding site, and that the general anesthetic was interacting with the binding of the lipid to the transmembrane domain. But this is the proposal we made, but it needs to be further demonstrated. Does not. So I showed the recent work which has been done on the different conformation of the lipids, and actually there is clearly some action of lipids on the conformation transition, late conformation transition of the activation process. So it has to be further studied.
But what would you say that targeting non-transmembrane domain, like extracellular, intracellular, is still of interest for drug design? So targeting other binding sites, not related to lipids at all. So do you still think that this is of interest?
There are of course binding sites in the transmembrane channel, which are channel blockers and which are allosteric, but in fact, we better to call them orthosteric, since we have two categories of orthosteric site, the acetylcholine site and the ion channel. So this is a channel blocker.
Yeah. So yeah, it would be interesting to look at. I may look at later on on channel blockers for glycine and for nicotinic acetylcholine receptors, because it's a typical approach. So yeah, it may be interesting reading. All right. Thank you very much. Thank you very much for the answers. So anyone else has any points, has any questions? Or otherwise, Zoe, we may close the meeting at some point.
Let me also personally thank Professor Changeux, Jean-Pierre. It was a great pleasure to host you. Always very insightful. Your knowledge is really shining in this field. I would like to thank you again for the lecture. Yes, I don't know if anybody else wants to add anything. If not, we hope to be able to reconnect again. And thank you very much again.
Thank you.
Thank you so much.
Congratulations, Zoe, and I hope that in the future, ALLODD will continue, because I think it's an opening field. It's at the end, and that's what I try to show. I think there are more and more and more openings in drug design, and I was happy to see in Greece actually that in the last day there were people from the industry being interested. My best wishes.
Exactly. It is really a field that is opening up new opportunities for all targets, for undruggable targets, for targets that were otherwise not accessible. So we have major interest from companies, from pharmaceutical companies. It was apparent indeed, as you mentioned in the Allostery Drug Discovery Conference. We had at least 1/4 of all the lectures were from industrial members, which meant that not only were they interested to participate, but also to share the results and share the knowledge and open up the field. And we certainly hope that the European Commission will feel this way that we are feeling as well, and that it will fund more projects in the field of allostery.
Thank you very much. Thank you.
Thank you very much.
Bye.
Bye.
Bye.